This genus is composed of a large or diverse group of bacteria that includes both animal and human pathogens, as well as saprophytes and plant pathogens. So very wide range po yung mga hosts na pwedeng i-infect ng Corynebacterium species, from humans to animals to plants. Now, Corynebacteria can be divided into non-lipophilic and lipophilic species. So those lipophilic Corynebacteria, they're often considered fastidious, and they grow slowly on standard culture media. Okay, so the cultures often must be incubated for at least 48 hours. So di pa ba yung mga usual na pathogenic bacteria na alam natin, within 18 to 24 hours lang po makakapag-observe na tayo ng colonies. Pero kung sinususpect natin na Corynebacterium yung isolate, then we should incubate the plates longer for up to 48 hours. However, if lipophilic nga po yung strains na present in the sample, then the presence of lipids or the addition of lipid sources in the culture media would induce their growth or would slightly speed up their growth. Now, on gram stain, Corynebacteria are slightly curved gram-positive rods with non-parallel sides and slightly wider ends. So, hindi po pantay yung itsura nila, and also mas malapad yung one end of the cell compared to the other end. So, this produces the term, this produces the appearance of being club-shaped. Okay, so another term for this would be coryneiform or diphtheroid, meaning diphtheria-like. Okay, so yun po yung gram stain morphology ng Corynebacterium species, as you can see here in this photo. So di ba po, hindi perfectly shaped yung bacilli. May isang side ng bawat bacilli na mas malapad compared to the other side. All right, so the most clinically significant pathogen in this genus is the Corynebacterium diphtheriae, and the disease that it causes is referred to as diphtheria. There are also non-diphtheria Corynebacterium species that can produce diseases in humans. So these include, but are not limited to, Corynebacterium amiculatum, Pseudodiphtheriticum, Pseudotuberculosis, JKM, Striatum ulcerans, and Urealyticum. So mamaya po, by the end of this segment, i-discuss rin po natin yung mga species na nanggaling dito that are considered non-diphtheria but are Corynebacterium. Kadalasan po sila ay mga opportunistic pathogens lang. Okay, so let's begin with Corynebacterium diphtheriae. So humans are the only natural host for C. diphtheriae. Hindi katulad ng ibang mga species belonging in this genus na wide range of hosts yung kaya nilang i-infect. For Corynebacterium diphtheriae, humans lang po yung natural host nila. Okay, so the bacteria is carried in the upper respiratory tract, and then this may spread by droplet inhalation or hand-to-mouth contact. Pwede rin naman po in cases of cutaneous diphtheria, this may also be spread with direct contact with lesions. Okay, so there is an effective toxoid vaccine that is available. So this is part of the trivalent diphtheria tetanus pertussis vaccine. So meron naman pong bakuna against Corynebacterium diphtheriae. So kasama na siya dun sa DTP. Kapag sinabi po nating trivalent, it means that in one vaccine, this may be used to prevent 3 different diseases. So again, that's what we mean when we say trivalent. So ito po, for the DTP vaccine, it may be used against diphtheria, tetanus, and pertussis. All right, let's now talk about the primary virulence factor of diphtheria. So, ayan po, the diphtheria toxin, this is the major virulence factor that is associated with C. diphtheriae. So this toxin is produced by strains of Corynebacterium diphtheriae that has been infected with a lysogenic bacteriophage, which carries the tox gene. Okay, so di ba po, na-discuss natin dati during the mechanisms of gene transfer. Some virulence factors may only be encoded by some bacteria if a bacteriophage infects them, and yung bacteriophage na to carries the gene sequence for that specific virulence factor. Okay? Now, non-toxigenic strains can be converted to tox positive by infection with the appropriate bacteriophage. So, hindi lahat ng Corynebacterium diphtheriae is automatically pathogenic and would automatically produce the diphtheria toxin. Okay, so kung na-infect lang po ito or nag-undergo siya ng gene transfer to inherit the tox gene, then dun lang po siya magiging capable of producing the diphtheria toxin. So the diphtheria toxin is composed of two fragments, fragment A and fragment B. So toxicity is caused by the ability of the diphtheria toxin to block protein synthesis in eukaryotic cells. So fragment A, this is responsible for cytotoxicity, and yung fragment B naman po, this binds to the receptors on the human cells, and this mediates the entry of fragment A into the cytoplasm. Okay, so mahalaga rin po yung fragment B, although it doesn't, or it's not the main component responsible for toxicity. Ito naman po yung component that is responsible for fragment A to enter the cytoplasm. Now, once it reaches the cytoplasm, fragment A disrupts protein synthesis. And when that happens, it could cause tissue necrosis and exudate formation. So this would lead to the production of pseudomembranes that would line the surfaces that it has infected. Okay, let's now look into the two forms of diphtheria. So the Corynebacterium diphtheriae causes two different forms of diseases in humans. So respiratory and cutaneous diphtheria. Let's first look into respiratory diphtheria. So the incubation period for this would average 2 to 5 days. So the illness begins gradually and is characterized by low-grade fever, malaise, and a mild sore throat. So mag-uumpisa po siya with non-specific symptoms. And the most common site of infection in the tonsils, or the most common site of infection would be at the tonsils or sa pharynx, as we can see here in this picture. Okay, so yung nakikita po natin na parang mukha lang nakalunok ng chewing gum yung pasyente, that's not actually gum. That is the pseudomembrane that has been produced by the bacteria when they were able to utilize their diphtheria toxin. So the organisms would rapidly multiply on the epithelial cells, and the toxigenic strains of Corynebacterium diphtheriae would produce the toxin locally, meaning kung nasaan po yung site of infection nila, dun lang po magpaproduce ng toxins. Now, this would cause tissue necrosis and exudate formation, triggering an inflammatory reaction. So this combination of cell necrosis, exudate formation, and inflammation would produce a tough gray to white pseudomembrane, which attaches to the tissues. This may appear on the tonsils and then spread downwards into the larynx and trachea. Okay, so ang problema po dito, there is potential for suffocation if the membrane blocks the air passage or if it is dislodged, usually as a result of sampling for throat cultures. So minsan po, ang pwedeng mangyari is that the infection would be left untreated, tapos lalaki lang ng lalaki yung pseudomembrane, titindi lang yung inflammation hanggang sa mabablock yung airways ng pasyente. Or ang mangyayari naman po could be during collection of the throat swab, matatanggal or madedetach yung pseudomembrane and malalaglag po siya papunta dun sa airways ng pasyente. So both of those scenarios could cause suffocation. Next is cutaneous diphtheria. So cutaneous diphtheria consists of non-healing ulcers with a dirty gray membrane, as we can see right here in the middle of this lesion. Okay? So parang ganun din po. Nag-form siya ng pseudomembrane dun sa area or dun sa specific spot that the bacteria has infected. So that was for cutaneous diphtheria, otherwise known as the Veldt sore or the Barcoo rot. Okay, on to laboratory diagnosis. Corynebacterium diphtheriae is highly pleomorphic, and this is also gram-positive. So it is a gram-positive bacillus that appears in palisades. So pag sinabi pong palisades, they are arranged side by side. So magkakatabi sila diagonally. Yun po yung meaning when we say palisade. So as we can see here in this photo, naka-point naman po dun sa arrow kung nasaan yung itsura ng mga cells that are arranged side by side in palisades. Sometimes they may also be seen in V or L formations. So yun po, V or L forms, example, one bacterial cell may be lying close to another bacterial cell at a right angle. So yun po yung tinatawag nila na V or L formation. As we can see right here in this zoomed in portion. So yan po yung example ng mga V or L forms. Now, the organisms often stain irregularly, especially if we would use methylene blue, giving them a beaded appearance as we can see right here. So for example, ito yung bacterial cell. Kung methylene blue yung gagamitin natin, there may be some areas of the cell that would not take up the stain. So magmumukha silang parang multiple beads, pero actually it's just one cell that did not stain properly. Next po, for the observation of metachromatic areas or the portions of the cell kung saan nagkaroon ng metachromatic granules, ayan, they would stain more intensely on other parts. And these metachromatic granules that is specifically produced by C. diphtheriae are called the Babes-Ernst granules. So kung nakita nyo po that the organism produces the Babes-Ernst granules, dapat po maisip nyo na agad is Corynebacterium diphtheriae. So these granules, they represent the accumulation of polymerized polyphosphates. So ang purpose po nito is if ever mapunta sila sa isang environment where there is not enough sources of or nutritional sources. Ayan po, at least meron silang parang baon through these metachromatic granules. And we may easily visualize them with the help of the uses methylene blue or the Albert stain, which makes use of malachite green. So itong third photo na ito po, this is an example of the Albert stain. All right, so on to the cultural characteristics of Corynebacterium diphtheriae. So C. diphtheriae is a facultative anaerobe. So it would grow best under aerobic conditions, pero kaya niya rin naman mag-survive under anaerobic conditions. And it has an optimal growth temperature of 37 degrees Celsius. Although multiplication could still occur within the range of 15 to 40 degrees Celsius. So wide range po yung temperature in which this organism may grow. So ayan po. On BAP or SBA, the organism can produce a very small zone of beta hemolysis. And then better growth is usually obtained on a medium that contains either blood or serum, such as the Loeffler serum or the pi agar. So characteristic microscopic morphology is demonstrated well when the organisms are grown on a Loeffler medium. So kung gusto po nating ma-observe yung or ma-observe ng mabuti or ma-encourage yung formation nila ng mga palisades nila tsaka yung iba pang mga characteristic morphology nila, then we may incubate them using the Loeffler agar. Next is the cysteine tellurite blood agar or CTBA. So this is a modification of the Tinsdale medium. So both of these culture media, they could either contain sheep red blood cells or bovine serum plus cysteine plus potassium tellurite. So CTBA is both a selective medium and a differential medium. Kaya po siya naging selective is because of potassium tellurite. Potassium tellurite inhibits the growth of many other non-coryneiform bacteria. So kahit na parehas silang Corynebacterium species, if they are non-coryneiform, may inhibit sila ng potassium tellurite doon sa culture media. And then, if the organism was able to grow on CTBA, they form black or brownish colonies. This is because of the reduction of tellurite. Okay? So, kung na-survive nga po nila yung potassium tellurite, pag na-survive nga po nila yung growth with the presence of potassium telluride, ayan po. Pwede rin nila tong i-utilize to produce black colonies. So ayan po. If ganito yung itsura ng colonies ng isolate nyo, then most likely this is Corynebacterium diphtheriae. Okay, so here on slide 10, let's look at some of the differential tests that we could do to differentiate Corynebacterium diphtheriae from other Corynebacteria. So first is the urease test. So the urease test, this detects the ability of the organism to produce urease or to be able to hydrolyze urea. So the hydrolysis of urea would lead to the formation of ammonia and carbon dioxide. Itong dalawang substances na ito together, they would turn the test environment into an alkaline environment. So magkakos po ng alkalinity or change in pH yung ammonia and CO2 na na-produce. Now, with the help of the pH indicator, which is phenol red, madidetect po niya yung change in alkaline. So a positive result is indicated by a pink or magenta colored change within the test media. So as you can see right here, pwede po tayong gumamit ng solution or pwede tayong gumamit ng agar slan. So ayan po, if nag-produce ng pink discoloration dun sa test media yung organism, then it is positive for urease test. Now, for Corynebacterium diphtheriae, magne-negative po siya sa urease test. Okay, so again, Corynebacterium diphtheriae would test negative for the urease test. Compared to other species, some of them may produce a positive result. Next po is the gelatin hydrolysis or the gelatinase test. So this detects the production of extracellular proteolytic enzymes. Yung mga agar po or yung mga culture media and other test media na ginagamit natin sa laboratory, they contain a solidifying agent, and solidifying agent po na yun is composed agar. But for the gelatinase test, is a substitute po natin yung agar component ng test medium to gelatin. So papalitan natin yung ng gelatin yung solidifying agent. Now, a positive result is indicated by partial or total liquefaction of the inoculated tube. So as we can see right here, parang nag-melt yung test medium na ginamit natin. So ito po, tube A, this is a positive result, and tube B, this is a negative result for the gelatinase test. So kung makikita natin, itong tube A parang nag-melt po yung test media. Pero dito sa tube B, ayan po, intact pa rin or same shape or there was no change that was produced in the gelatin stab. So again, ang Corynebacterium diphtheriae po, it would produce a negative result. And then some other Corynea species might produce a positive result. Okay, let's now talk about toxigenicity testing. So the LEC test is a form of toxigenicity testing. Kasi po, the identification of an isolate as Corynebacterium diphtheriae does not automatically mean that the patient has diphtheria. Kasi nga po, di ba, nabanggit natin kanina, some diphtheria species may be negative for toxin production. So yung iba namang pong diphtheria species, they may only be commensals or normal flora lang sila. So the diagnosis of diphtheria is established by demonstrating if the isolate also produces the diphtheria toxin. So with the help of the LEC test, each organism is straight in a single straight line parallel to each other. And approximately there should be about 10 millimeter distance between each inoculum. Okay, so after that, a filter paper strip that is impregnated with diphtheria antitoxin is laid along the center of the plate on a line that is at a right angle to the inoculum lines. Ayan po. So tignan natin to. The plate is incubated at 35 degrees Celsius and examined after 18 to 24 to 48 hours. So, titingnan natin siya in intervals of time. So, we would start at 18 hours and then kung wala pa, we'd check again after 24 hours. And then final results would be reported after 48 hours. So, ayun po, tingnan natin tong example na to. Usually, we would first include a positive control. So, ayan po. Yung positive control, it is probably, ito po, yung inoculum number 1. So, kung nakikita natin, dito po sa gilid nung, dun sa gilid ng inoculum, nakatabi ng filter paper, there were white precipitin lines that were formed, and they are usually located at a 45-degree angle from the line of growth. So yung mga nakikita po natin na parang nakatagilid na letter V, yun po yung tinatawag natin na precipitin lines. So these precipitin lines, they are produced after mag-react nung diphtheria toxin na produce ng bacteria versus yung diphtheria antitoxin na kino-contain nitong filter paper dito sa gitna. Okay? So this one, ito po yung positive control. So if magpo-positive yung organism, dapat po ganito yung itsura niya. If we would look at inoculums number 2 and 3, hindi sila nag-form ng precipitin line. So most likely negative po itong dalawang isolates na to. And then if we would look at inoculum number 4, ayan po, kahawig niya yung positive control, which is inoculum 1. So meron din siyang mga lines na na-produce sa gilid. So most likely we could say that sample number 4 is the one that is positive for the production of diphtheria toxin. So yeah, that was for the LEC test, which is a toxigenicity testing. Dahil po, again, not all Corynebacterium species or not all C. diphtheriae can produce the diphtheria toxin. So hindi po lahat ng C. diphtheriae isolates would automatically mean na it was the causative agent of the patient's disease. Okay? Next is the Schick test. So the Schick test is a skin test used to determine the susceptibility of an individual to diphtheria. So this procedure involves the intradermal introduction of a small amount of diphtheria toxin into the arm of the individual who is suspected of having diphtheria. Okay, so a positive result is indicated by redness and swelling on the site of injection. So parang parehas sila ng konsepto nung skin test for tuberculosis. Ayan. So again, this is Schick test for Corynebacterium diphtheriae. Corynebacterium diphtheriae also has 3 different biotypes. Okay? Alright, 3 different biotypes. So, ang difference lang naman po nila is kung ano yung itsura ng colony morphology na mapoproduce nila. Okay? So, first up is intermedius. The intermedius biotype produces small, flat, dry, grayish colonies as we can see right here in the first photo. Okay? So kapag mitis naman po yung biotype, they would produce small, black, fried egg appearing colonies. As you can see right here in this second photo. So kaya po sila tinawag na fried egg colonies kasi, di ba, fried eggs, yellow yung gitna, tas white yung labas. So parang ganun daw po yung itsura ng mitis biotype. So meron siyang black sa gitna, tas white yung labas. Okay, so that's for the mitis biotype of Corynebacterium diphtheriae. Last one is the gravis biotype. So they produce large, flat, dark gray colonies that have a daisy head appearance. So mukha daw po silang flowers or daisy heads kasi solid yung gitna tapos mukhang dispersed yung paligid ng colonies. So yan, that was for the gravis biotype of Corynebacterium diphtheriae. So ayun po, these 3 biotypes, most likely ma-observe natin yung differences nila based on ano ang itsura ng colonies nila. Let's now talk about all the other Corynebacteria or yung mga Corynebacterium species po that are non-diphtheriae. Okay, so let's start with Corynebacterium urealyticum. So this is one of the most frequently isolated and clinically significant Corynebacterium species, right next to or second to C. diphtheriae. Okay, so this is usually a urinary pathogen, so most likely ma-observe natin to in cases of UTI, and it is urease positive. So nabanggit natin kanina, di ba? Differential tests for Corynebacterium diphtheriae versus other Corynea species, ayan, is the urease test. So kung negative yung C. diphtheriae, magpa-positive naman po ang Corynebacterium urealyticum. So as the name implies, di ba po, urealyticum, it is urease positive. Next is Corynebacterium jacqueum. So this is a common cause of diphtheroid prosthetic heart valve endocarditis. So meron din pong species ng Corynebacteria na kayang magdulot ng prosthetic heart valve endocarditis. So that's for Corynebacterium jacqueum. And then third one is Corynebacterium pseudodiphtheriticum, otherwise known as the Hoffman bacillus. So this is a normal flora of the human nasopharynx, and diseases associated include respiratory infections, wound infections, and UTI, specifically among immunocompromised patients. Okay? So nabanggit ko po kanina, 'di ba, na being able to isolate Corynebacteria from the patient's throat samples does not necessarily mean that the organism that the organism was able to cause diphtheria, or it is diphtheria that that the disease, or yun yung disease na meron yung pasyente. Okay, so ayan po. This is because Corynebacterium pseudodiphtheriticum is considered as a normal flora of the nasopharynx. So, ayun po, hindi natin i-establish agad yung clinical significance niya right away. Kasi nga po, considered siya as a normal flora. Now, parang i-consider lang natin yung clinical significance niya if immunocompromised po yung pasyente. Okay? So again, kasama siya sa mga urease positive na Corynebacterium species. All right, let's proceed to Listeria monocytogenes. So Listeria monocytogenes is widespread in the environment and has been recovered from soil, water, vegetation. It is also found in animal products such as raw milk, cheese, poultry, and some processed meat. Also, it has been known to cause illness in many species of both wild and domestic animals. So that includes sheep, cattle, horses, and even pets such as dogs and cats. And it can be isolated from both human and asymptomatic carriers. So they're basically everywhere. The optimum growth temperature for Listeria monocytogenes is at 30 to 35 degrees Celsius. But because Listeria monocytogenes has the special capability of growing at 4 degrees Celsius, dito po na-develop yung cold enrichment method or yung cold enrichment technique. Okay, so the cold enrichment method may be used to isolate organisms from a polymicrobial clinical sample. So again, pag sinabi nating polymicrobial, this means that the sample contains multiple organisms. So kung gusto nating malaman if kasama doon sa mga organisms na yun yung Listeria monocytogenes, we could incubate this at 4 degrees Celsius. Now, 4 degrees Celsius usually po inhibitory na siya to other organisms dahil masyado na itong malamig para sa kanila. Hindi ito yung optimum growth temperature nila. But yun nga po, ang special capability ng Listeria monocytogenes is that it can grow at 4 degrees Celsius. Okay? Now, in wet mount preparations, Listeria monocytogenes exhibits a tumbling motility or an end-over-end motility when this is viewed microscopically, as shown here in this GIF. So, ayan po. Yun yung end-to-end motility. Pag sinabing end-to-end motility, if for example, this is the bacterial cell, this is one end of the cell and this is the other. Pag sinabi pong end-to-end, yung movement niya, pag ganito talaga. So, mag-move siya from one end to another. So that is the end-to-end motility. of Listeria monocytogenes. Okay, this is because they have peritricous flagella. Now, with regards to the virulence factors of Listeria monocytogenes, they have hemolysin, they have catalase, superoxide dismutase, and also phospholipase C, and a surface protein P60. So this protein P60, this induces phagocytosis through increased adhesion and penetration into mammalian cells. So hindi katulad ng ibang mga um, pathogenic bacteria na ang virulence factor nila is to avoid phagocytosis, ang Listeria monocytogenes po, it induces or it encourages phagocytosis thanks to its P60 protein. And then another virulence factor is the listeriolysin O. So this damages the phagosome membrane, effectively preventing the killing of the organism by macrophages. So if you would remember yung discussion natin on the four main steps of phagocytosis, di ba kasama po dun yung formation of the phagosome? Yung phagosome is the vacuole that contains the engulfed organism. Ang special capability po ng Listeria monocytogenes is that it is able to get out or destroy the phagosome with the help of listeriolysin O. So, wala na rin pong pakinabang if the phagosome would fuse with the lysosome kasi wala na dun sa loob yung Listeria cell. Okay, let's now talk about listeriosis. So studies have indicated that the ingestion of contaminated food with subsequent spread through the intestine is the primary mode of transmission for listeriosis. So ayan po, ito yung mga different forms of listeriosis. First is maternal. So in pregnant women, listeriosis is most commonly seen during the third trimester. So Listeria monocytogenes is responsible for the spontaneous abortion or stillbirth of neonates. So kapag sinabi pong stillbirth, this is the death of a baby before or during the delivery. So ipapanganak po yung bata na patay na. Next is neonatal listeriosis. There are two forms of neonatal listeriosis, early and late onset. Early onset listeriosis results from an intrauterine infection that can cause illness shortly after birth. So most likely after a few hours lang po, mapapansin na natin yung clinical presentation na to. So the result is most often sepsis. So again, kapag early onset po, just a few hours after birth, ma-observe na yung manifestation. And the manifestation is usually sepsis. Kapag naman po late onset, the disease occurs several days to weeks after birth. So, hindi agad-agad. It might take days, it might take even weeks bago tayo makapag-observe ng symptoms. Okay? So, the affected infants are generally full-term and healthy at birth. Pero po, ayun nga, after a few weeks, the disease would most likely manifest itself as meningitis. Okay? So, yun po yung difference between early onset neonatal listeriosis and late onset neonatal listeriosis. Kapag early onset po, sepsis yung mapoproduce. Kapag late onset, meningitis. Now, let's look into the listeriosis for immunocompromised individuals. Invasive listeriosis is commonly observed in persons who are immunosuppressed and also in older adults, and particularly those patients that are receiving chemotherapy. So the most common manifestation of listeriosis in immunocompromised people are CNS infections and endocarditis. Okay, tingnan na po natin yung laboratory diagnosis of Listeria monocytogenes. So, Listeria monocytogenes grows well on SBA and also on chocolate agar and other culture media or broth such as the brain heart infusion medium or the thioglycolate broth. So, bali, hindi naman po fastidious ang Listeria. Hindi naman po siya mahirap i-culture across many different routine culture media. Their colonies may be small, round, smooth, and translucent. Excuse lang po, may insekto po kasi. Sorry, narinig ko po kasi may zzz sa likod ko, tas may insekto. Sorry. Now, as I was saying, the colonies of Listeria monocytogenes may be small, round, smooth, and translucent. Okay, so they are surrounded by a very narrow zone of beta hemolysis, and this may only be visualized if the colony is removed or scraped off. Okay, so nagpaproduce po sila ng beta hemolysis. The problem is initial inspection might mislead us to think that they are non-hemolytic. Pero yun pala, if we were to scrape off some colonies, or if we would look under the colonies, ayan po, doon lang natin makikita yung beta hemolysis nila. Ayan, in wet mount preparations, Listeria monocytogenes exhibits stumbling motility or the end-over-end motility. So when viewed microscopically, napakita ko na po yun kanina, di ba? Yung end-over-end motility ng Listeria. Now, another way that we could observe their motility is by inoculating them in a motility medium, or the SIM, or the sulfide indole motility medium. So with this culture media, they would produce a characteristic umbrella pattern. Okay, so this is usually seen if the organism has been incubated at temperatures of 22 to 25 degrees Celsius. Okay, so kapag nakita nyo po, sinabi that the organism was able to produce an umbrella-shaped motility, ayan po, dapat maisip nyo na agad is Listeria monocytogenes. Okay? Next, Listeria produces a positive CAMP reaction. So pwede rin po natin gawin yung CAMP test for Listeria monocytogenes. They would produce a block-type hemolysis, or somewhat a rectangular pattern, as we can see in this example. So if i-compare po natin yan sa CAMP reaction ng Streptococcus, di ba po sa Streptococci, they produce a triangular or arrowhead shaped hemolysis. Dito naman po, same din yung konsepto, no? Listeria monocytogenes would produce the CAMP factor. This would react synergistically with the beta lysine of Staphylococcus aureus and would produce enhanced hemolysis. Ang magkaiba po sa kanila is yung shape. So for Streptococci, kung ang shape ng hemolysis niya during the CAMP test is arrowhead, dito naman po for Listeria monocytogenes, ang shape ng result niya is block type, meaning hugis rectangular siya. Okay, let's now look into the differential tests that we could do if we would like to differentiate Listeria monocytogenes compared to Corynebacterium species. Okay, so first up is hippurate hydrolysis. So if the bacteria produces the enzyme hippuricase, this can hydrolyze hippuric acid to glycine and benzoic acid. Yung glycine po na na-produce ng reaction na yun, this can be detected after the addition of ninhydrin, which would form a purple colored product, as we can see right here. So positive po for hippurate hydrolysis ang Listeria, pero negative ang Corynebacterium species. Next is the bile esculin hydrolysis. So nabanggit na rin po natin to kanina, di ba, on chapter 15. So if the bacteria is capable of growing in the presence of increased salt concentration plus 4% bile, and it is able to hydrolyze esculin to esculitin, ayan po, the esculitin will then react with the ferric ions that are present in the culture media. Okay, so this would form dark brown or black precipitates, as we can see here in these examples. So again, magpa-positive po dito ang Listeria monocytogenes, pero negative po ang Corynebacterium species. Okay, let's now move on to the non-spore forming, non-branching, pero catalase negative bacilli. So kanina po, di ba, same lang din yung descriptions except that Corynebacteria and Listeria are catalase positive. All right, so first up is Erysipelotheryx. There are 3 species in the genus Erysipelotheryx, which includes Erysipelotheryx rhizopathiae, tonsillarium, and inopinnata. So erysipelosis crucopathiae is the only species in this genus that is known to cause diseases in humans. So it is gram-positive and catalase negative. It is also non-spore forming, and this is a pleomorphic rod. So although established na bacilli yung shape nila, minsan po irregular yung itsura ng bacilli. Now, human cases typically result from occupational exposure. Individuals whose work involves handling fish and animal products are most at risk. So the usual route of infection is through the cuts or scratches on the patient's skin. All right, so erysipelosis crucopathiae produces 3 types of disease in humans. First up is erysipeloid, which is a localized skin disease. Next is septicemia, which is often associated with endocarditis. And last one is a generalized diffuse cutaneous infection. Ang pinakamahalaga po natin kailangan tandaan dito is erysipeloid. So this is the most common infection that is caused by erysipelosis crucopathiae in humans, and this is a localized skin infection that resembles streptococcal erysipelas. So medyo magkamukha po yung lesions na napoproduce during erysipeloid and erysipelas. So tandaan lang po na magkaiba sila ng causative agent. Ang erysipelas po is caused by streptococci, and ang erysipeloid po is caused by erysipelosis. Okay, so the lesions are usually seen on the hands or fingers because the organism is inoculated through work activities. So, di ba, nabanggit natin that erysipelosis crucopathiae is acquired through occupational exposure. So the incubation period is approximately 2 to 7 days, and the infected area is painful and swollen, as you can see right here. Meron din pong characteristic motility pattern ang erysipelosis crucopathiae sa gelatin stab or gelatin medium. So erysipelosis would produce a test tube brush-like growth, as we can see here in this second photo. So ito po yung tinatawag na test tube brush pattern, which is produced by erysipelosis crucopathiae. Okay? So dalawa na po yung nakita natin, no? For Listeria, it's an umbrella pattern. For erysipelosis, it's a test tube brush pattern. Next is Arcanobacterium hemolyticum. So the primary human pathogen in this genus is Arcanobacterium hemolyticum, which is formerly known as Corynebacterium hemolyticum. So the pharyngitis that is caused by Arcanobacterium hemolyticum can be mild or severe, and this is often clinically indistinguishable from pharyngitis caused by beta hemolytic streptococci. So kahawig niya daw po yung clinical presentation ng strep throat. Okay, so upon culturing, Arcanobacterium hemolyticum produces small colonies on SBA that demonstrate a narrow zone of beta hemolysis after 24 to 48 hours of incubation, which is similar in appearance to that of beta hemolytic streptococci. So maliban sa clinical presentation, magkahawig din sila ng colony morphology ng ibang beta hemolytic strep. And frequently, a black opaque dot is observed on the agar when the colony is scraped off. Or there may also be pitting of the agar beneath the colony. Okay, so if i-scrape off natin or tatanggalin natin yung mga colonies from the agar, pwede nating tingnan yung ilalim. So either magkakaroon ng black dot sa gitna or magkakaroon ng pitting of the agar. When we say pitting of the agar, magkakaroon ng maliit na butas. So mabubutas yung agar dun sa area kung saan nag-grow yung colony. Okay? Ngayon, pwede rin po natin gawin yung camp test sa Arcanobacterium hemolyticum. So on camp test, this would exhibit a reverse camp reaction or a camp inhibition due to phospholipase D. Okay? So, di ba po, kanina in-explain ko yung konsepto ng camp test. An organism that produces the camp factor would synergistically react with the beta lysine that is produced by Staphylococcus species. This would then lead to enhanced hemolysis. So kabaliktaran po nito yung result ng Arcanobacterium hemolyticum. Ang mangyayari is yung beta lysine that is produced by Staph aureus, it would react with the phospholipase D that is produced by Arcanobacterium hemolyticum, and it would inhibit or lessen the capability to produce hemolysis. Okay? So ayan po. If we would look at this photo to the lower left, ayan po, ito yung example dito sa kanan ng plate. Ito yung example ng isang positive camp test. So, dun sa area kung saan nag-meet yung dalawang streak lines, na-enhance yung hemolysis. Kabaliktaran nito yung na-produce ng Arcanobacterium hemolyticum. So, dun sa mga areas kung saan nag-meet yung Staph aureus and yung Arcanobacterium hemolyticum streak, na-lessen yung capability ng Staph aureus to produce hemolysis. So ito po yung tinatawag na reverse camp. Okay, next is Gardnerella vaginalis. So these are short pleomorphic gram-positive rods or cocobacilli that would often stain gram-variable or gram-negative. Okay, so Gardnerella vaginalis, although this has been established as a gram-positive type of bacteria, yung cell walls niya po kasi, they have a thinner peptidoglycan layer. Okay, so compared sa mga regular na gram-positive organisms, mas manipis po yung peptidoglycan layer ng Gardnerella vaginalis, which is why mas kakaunti yung surface area na pwedeng pagkapitan ng primary stain, and also may possibility rin na mas madali silang ma-decolorize during the decolorization step of gram staining. Okay? So Gardnerella vaginalis is primarily known for its association with bacterial vaginosis. So for many years, or in the past, Gardnerella vaginalis was thought to be the only causative agent of bacterial vaginosis. However, further studies were able to confirm that bacterial vaginosis is actually a polymicrobial disease. So kasama lang po dito, or isa lang dito, yung Gardnerella vaginalis. But there are other organisms such as Prevotella or Peptostreptococcus or Porphyromonas and Mobilionicus that could work together to produce bacterial vaginosis. Okay? So this is characterized by malodorous discharge and a vaginal pH that is greater than 4.5. So papunta na po ng alkaline yung pH ng vaginal area ng pasyente if they have bacterial vaginosis. So this generally results from a reduction of the lactobacillus population in the vagina, followed by the increase of vaginal pH. So di ba po, na-discuss natin during our discussion on commensal bacteria and yung mga reasons as to why they colonize a specific area in the human body. So yung mga lactobacillus po, they are considered as commensals in the vaginal area of the patient kasi po, or of regular, normal, healthy humans kasi po, they are able to utilize substances from that area. So their utilization of those substances from that area produces an acidic pH, which is detrimental to all other organisms. So ang mangyayari po in bacterial vaginosis, kokonte yung population ng lactobacillus and aangat yung population ng Gardnerella vaginalis and other bacteria that is associated to BV. Okay? So yun po. After that, tataas na rin po yung pH ng area and it would produce an alkaline pH. All right, so the medium of choice for Gardnerella vaginalis is the human blood bilayer twin agar, or the HBT agar. So when cultured on human blood, colonies of Gardnerella vaginalis would produce beta hemolytic, small, gray, and opaque colonies, as you can see right here in this top right photo. Okay, next is gram staining. Gram staining of vaginal secretions is generally regarded as the reference method for the diagnosis of bacterial vaginosis. So we should be able to observe clue cells. Clue cells are large squamous epithelial cells with gram-positive or gram-variable bacilli and coccobacilli that are clustered on the edges of the cell, as we can see right here in this lower right picture. Okay, minsan po, hindi lang basta sa edges nakakoncentrate yung bacteria. Minsan sobra yung overgrowth nila that the epithelial cell is totally covered in bacterial cells. Ayan po. So if we would compare, ito yung itsura ng isang clue cell and ito po yung itsura ng normal, healthy vaginal epithelial cells. Okay, so next is the whiff test. Okay, so yung whiff test naman po, after the addition of 10% potassium hydroxide, a fishy odor would develop, and this indicates that the infection is probably bacterial vaginosis. Okay, so ulitin po natin yon. 10% KOH plus vaginal secretions from the patient, if they would produce a fishy odor, then yun po, most likely bacterial vaginosis yung diagnosis ng pasyente. Okay, let's now proceed to non-spore forming, branching, aerobic actinomycetes. Okay, so starting off with the Nocardia species, Nocardia are aerobic, branched, gram-positive bacilli. Aside from being gram-positive, Nocardia species are also partially acid-fast, meaning they're able to retain the primary stain only after or only if we would use a weak acid as a decolorizer in the acid-fast staining process. So dito po, pumapasok yung modified acid-fast staining. Papalitan natin yung decolorizer, yung usual decolorizer with a weaker form of acid. Okay, so magpa-positive po for modified acid-fast staining ang Nocardia species. Okay, so maliban sa gram-positive siya, partially acid-fast din siya. Alright, so the colony and microscopic morphology, as well as the types of infections that is caused by Nocardia species may resemble those of fungal infections. So microscopically, macroscopically, and based on clinical presentations, pwede po nating mapagkamalan yung Nocardia species as fungal pathogens. Okay, so Nocardia species can be recovered on standard non-selective media, meaning hindi naman siya fastidious. Kaso lang, it may take 1 week or longer for colonies to show up. So, although hindi siya fastidious kasi wala siyang additional nutritional requirements na kailangan, it is a slow grower. Kasi approximately 3 days to 1 week po bago tayo makakita ng colonies. Now, the colonies of Nocardia species might have a chalky, matte, or velvety or powdery appearance, and they may have various pigmentations. So they can be white or yellow or pink or orange or tan or gray. And what they do have in common is they can have a dry, crumbly appearance similar to breadcrumbs. So ayan po, yun yung characteristic description for Nocardia species. They could produce or they could have a breadcrumb appearance. So ito po yung itsura ng Nocardia species on culture, as you can see here in this third photo. Ayan po, itong first photo, this is an example of the branching of Nocardia species. So yung mga cells niya, they may be arranged in a way na mukha silang magkakaduktong and they are forming branches. So ito po yung itsura nila on gram staining. If we were to perform partial acid fast staining or modified acid fast staining, ito naman po yung magiging itsura nila in this picture in the middle or the second photo. Ayan, so yung mga red na cells po, yan yung Nocardia cells. So generally, infections caused by Nocardia occur in immunocompromised patients. So we could say that this is an opportunistic pathogen. So the most commonly encountered species are Nocardia brasiliensis, which causes actinomycotic mycetoma, and also Nocardia cereosigorgica, which could cause bronchopneumonia. Okay, so ayan po, Nocardia species, they don't require any specific growth factors. and they would grow well on most common non-selective laboratory media if they are incubated between the temperatures of 22 to 37 degrees Celsius. Although, yun nga po, they are slow growers, so it would take probably like 3 to 6 or 7 days before organisms or before colonies would be observed. All right, let's talk about all the other actinomycetes that are also considered as opportunistic pathogens. So first up, aerobic actinomycetes of clinical importance. They belong to the genus Actinomadura. So ang mga common isolates po dito is Actinomadura madure and Actinomadura peleateri. So these can cause mycetomas, which are identical to those that are caused by nocardia. And both their microscopic and macroscopic morphology kahawig din po ng nocardia species. So these organisms can be identified using metabolic variations. So dahil magkamukha sila microscopically and macroscopically, ang gagawin natin, magbabiochemical testing tayo para ma-differentiate natin yung Actinomadura versus nocardia species. Okay? So one test that we could perform is the carbohydrate utilization test. So sa carbohydrate utilization test po, kung ang gagamitin natin is cellubiose and xylose, magpa-positive po ang Actinomadura, meaning kayang i-utilize ng Actinomadura yung cellubiose and yung xylose. Pero po, for nocardia, they can't do this. Okay? So nocardia species will not produce any form of acid with those two carbohydrates. Next is Streptomyces species. They are primarily saprophytes that are found in the soil or outdoor environments, and they may also resemble other actinomycetes. So isolates from the genus may be or may need to be identified by reference laboratories. So very rare lang po kasi yung infections with Streptomyces, and usually nga po environmental contaminants lang sila because they are saprophytes. So they prefer to inhabit the soil or other external environment. So kung gusto po talaga nating i-check if Streptomyces nga yung causative agent ng disease, then we would have to forward our sample to a reference laboratory. Next po is Gordonia. So for Gordonia, these are aerobic catalase positive or gram variable aerobic catalase positive, gram positive to gram variable organisms, and they may also be partially acid fast. Okay, so again, kahawig na naman niya yung nocardia. They may be distinguished from the genus Nocardia by their ability to reduce nitrate. So kung gusto natin i-differentiate yung nocardia versus Gordonia, we could do the nitrate reduction test. So magpa-positive yung Gordonia and negative ang nocardia. Okay? Next is Rhodococcus equi. So Rhodococcus equi, this is the most common human isolate in the said genus, and this is found in the soil and can cause respiratory tract infections in animals. Now, human infections are quite rare, although there is an increased incidence among immunosuppressed patients. So again, this is an opportunistic pathogen. Contact with farm animals and their infected feces is an important risk factor. Okay, so upon gram staining, Rhodococcus equi may demonstrate filaments and branching na kahawig ulit ng Nocardia species. Rhodococcus equi may also be partially acid fast. Now, again, it's something that is the same with Nocardia species. Now, the key feature for identification of Rhodococcus equi is its production of salmon pink colonies. Ayan po. So, microscopically, magkahawig yung Nocardia and yung Rhodococcus. Pero, macroscopically, ma-differentiate natin sila because of the production of salmon pink colonies, as you can see right here. So, kapag nakita nyo po na salmon pink yung isolate, then most likely po, Rhodococcus equi yung kino-contain ng clinical sample natin. Last one is Tropharema whipple. Okay, Okay, so Tropariema whiffle is the causative agent of Whipple disease. So Tropariema whiffle has been detected in human feces, saliva, and gastric secretions. And this is also ubiquitous in the environment. So if Whipple disease is left untreated, this is actually a fatal disease. So this includes symptoms of diarrhea, weight loss, malabsorption of nutrients, arthralgia or painful joints, and abdominal pain. Eventually, this could progress to neurologic and sensory changes. Now, for the diagnosis of the Whipple disease, this is best made by microscopic examination of endoscopic biopsy specimens. And ang staining method po would be the periodic acid shift staining. Okay, so hindi culture, hindi biochemical testing yung preferred method to diagnose the Whipple disease. Rather, we should perform periodic acid shift staining using endoscopic biopsy samples. All right, let's proceed to the last segment of this discussion. Let's talk about the Bacillus species, primarily yung Bacillus anthracis and Bacillus cereus. Okay, so for the general characteristics of Bacillus species, the members of the genus Bacillus may stain gram-positive or gram-variable, and they are aerobic or facultative anaerobes that can form endospores. Okay? So yung survival po of Bacillus species in nature is aided by the formation of endospores, which are resistant to conditions that are usually harsh or too harsh for the vegetative cells. Okay? So kung ang purpose po ng endospores is kung mapupunta yung organism sa isang environment that is unfavorable or hindi favorable yung growth conditions, ayan po, at least kahit mamatay yung vegetative cell, magsusurvive yung endospores. Now, all Bacillus species are motile with peritrichous flagella except for Bacillus anthracis and mycoides. So, among these two, ang clinically significant lang naman po is Bacillus anthracis. And upon gram staining and microscopic examination, they are large and very rectangular. So, we have the description boxcar shape. Okay, so yung mga boxcar, yung mga shipping containers, ayan po, kahawig daw nila yung mga boxcars kasi very rectangular yung shape nila. Right? And of course, they also have their characteristic unstained central spores. So these are endospores that are located in the middle of each bacilli. Okay, now let's talk about the virulence factor of Bacillus anthracis. Okay, so the virulence of Bacillus anthracis depends on a glutamic acid capsule plus a 3-component protein exotoxin. Okay. So, ayan po, the capsule, which protects the organism from phagocytosis, is a polypeptide of D-glutamic acid. So, yung capsule niya is composed of glutamic acid. Before we proceed, recall time. Recall time muna po from our chapter 15 discussion. If the capsules of Bacillus anthracis are composed of glutamic acid, ano naman po yung composition ng capsules of Streptococcus agalactiae? Sige po, if anybody remembers yung na-discuss natin kanina, ayan. Again, what is the composition of the capsules of Strep agalactiae? Glue acid din po ito. All right, the correct answer is sialic acid, as shown here in this pop-up screenshot. So both yung Strep agalactiae and Bacillus anthracis, capsule po nila yung primary virulence factor nila. And difference nun is that The capsules of Strep agalactiae is composed of sialic acid, while the capsules of Bacillus anthracis are composed of glutamic acid. Okay? Okay, so ayan po. Meanwhile, ito naman po mga nabanggit na protein exotoxins. The anthrax toxin consists of 3 proteins. So, 1 protective antigen, 1 EF, and 1 LF protein exotoxin. Each of them individually is non-toxic. Okay? So, non-toxic po ito kung magkakahiwalay sila. But together, together they can act synergistically to produce damaging effects. So, ayun po. As mentioned earlier, they may grow aerobically or anaerobically since they are facultative anaerobes, and they could also grow in acidic environments. So sa mga environment na less than 6 yung pH, and also they are halophilic, meaning mabubuhay po sila. They are able to withstand up to 7% salt concentration. And also, ayan po, all Bacillus species are non-motile except for Bacillus anthracis and Mycoides. Pero yun nga po, between the two of them, Bacillus anthracis lang naman po yung clinically significant. Okay? This bacteria is also categorized as a Category A bioterrorism agent by the Centers for Disease Control and Prevention. Okay, so meron pong tatlong categories of bioterrorism agents. And ang Category A po, this is the highest priority among all of them. So these include organisms that pose a risk to national security because they can be easily disseminated from person to person. They have high mortality rates. They might cause public panic, and they require action for public health preparedness. So Bacillus anthracis po was able to meet all of these 4 descriptions, kaya po naisama siya sa Category A, bioterrorism agents. Okay, let's now talk about the 3 clinical forms of anthrax. So humans are infected primarily as a result of accidental or occupational exposure to animals or animal products that is contaminated with Bacillus anthracis. So first up is cutaneous anthrax. Okay, so cutaneous anthrax, this begins as a small pimple or papule that appears at the site of inoculation 2 to 3 days after exposure. Okay, so ayan po. Ayan. After that, a vesicle would develop, or a ring of vesicles would be formed around this papule. And then those vesicles would coalesce or would join together to form an erythematous ring, as we can see here in this photo. So yung parang red edges, yun po yung vesicles. And then there would be a small dark area that appears in the center of the ring, and eventually this would ulcerate and dry and form a depressed black necrotic central area. So ito po yung tinatawag na black eschar, as shown here on the top right picture. Okay, so this eschar is normally 1 to 3 centimeters in diameter, or approximately 1 inch in diameter. And it may be more extensive, and this would only begin to heal after 1 to 2 weeks. So medyo matagal po yung healing process ng black eschar. Next is inhalational anthrax, or the pulmonary anthrax, otherwise known as the wool sorter's disease. Okay, so the wool sorter's disease is acquired when the spores are inhaled into the pulmonary parenchyma. So the infection begins as a non-specific illness consisting of mild fever, fatigue, and malaise 2 to 5 days after exposure to the spores. So it resembles an upper respiratory tract infection, such as those that are seen in colds or flu. So magsisimula po siya as very non-specific. So mukha lang may sipon or mukha lang may trangkaso yung pasyente. Now, this is then followed by a sudden severe respiratory distress. So dito na po papasok yung capability ng organism to be a category A bioterrorism agent. Kasi pagdating dun sa severe phase or dun sa respiratory distress, magiging fatal na po yung infection. So respiratory problems include dyspnea, which is the shortness of breath or difficulty breathing. And then also there would be cyanosis, which is the bluish discoloration of the skin due to lack of oxygen in the blood. And third one is pleural effusion. So pleural effusion is the buildup of excess fluid between the layers of the pleura and the outside of the lungs. So in simpler terms, magkakaroon po ng tubig sa paligid ng baga ng pasyente. Okay? So all of these would eventually progress to disorientation, coma, and death. Next is gastrointestinal anthrax. So this occurs when the spores are inoculated onto a lesion on the intestinal mucosa after ingestion of the spores. So the symptoms of gastrointestinal anthrax include abdominal pain, nausea, anorexia, and vomiting. Minsan po, bloody diarrhea could also occur. Ngayon, meron pa pong isa pang form of clinical anthrax that has been recently recognized. So this is known as the injectional anthrax. So this has emerged more recently and is recognized as an additional form of clinical infection. Hindi ko na po naisama dito. Okay, so infection results from the direct injection of spores into the tissues, generally during the administration of drugs of abuse. So common po yun for IV drug users. Okay, so again, that was for injectional anthrax. Hindi ko na po nailagay dito, pero kasama po yun dun sa book ninyo. On to the laboratory diagnosis. If the clinical sample is suspected to contain Bacillus anthracis, then it is strictly advised that we process this at a BSL-3 laboratory. And also, ayan po, ang itsura nila after gram staining, nabanggit na natin kanina, they are boxcar shaped with an unstained central spore. Pero another method of staining that we could use is the Schaefer-Fulton endospore staining, which makes use of malachite green. Okay, so as shown below, ayan po, ang primary stain dito is malachite green. So yung mga endospores po, or yung gitna ng cell, they would have a green discoloration, or green discoloration, green staining reaction. Okay, tas yung labas po ng cell, ayan, yung labas po ng cell, yun yung magiging kulay red. Kasi po, ang secondary stain dito sa Schaefer-Fulton is okay, let's now talk about the colony morphology of bacillus. So on BAP or SBA, they are non-hemolytic, large, and they may be gray, flat with an irregular margin because of outgrowths of long filamentous projections. Okay? So kapag sinabi pong filamentous projections, yung gitna ng colonies, solid sila. Pero yung labas ng colonies, medyo feathery yung edges nila. So yun po yung tinatawag na filaments. Because of this appearance, ayan po, this was given a term of medusa head. Okay, so yun po yung ibig sabihin when we say medusa head. Solid yung gitna, tapos po yung outer edges, ayan, they are filamentous. Take note that they are also non-hemolytic. And ito naman po yung itsura nila on cap. Okay, so ayan po, medyo magkahawig lang din naman sila. There might be differences only because of some strains. So some strains may have varying morphologies. Next is the egg yolk agar. Ayan, the egg yolk agar is used to determine if Bacillus anthracis has the ability to produce lecithinase. So if they do, there would be an opaque zone of precipitate that is produced in the immediate surrounding of the colonies. Okay. Next is the phenylethyl alcohol agar and also the polymyxin lysozyme EDTA thalassacetate medium or the PLET medium. Okay, so halos lahat ng ingredients in-enumerate nila dun sa pangalan ng medium. So yung PEA po and also yung PLET medium, this could be used to isolate Bacillus anthracis from from samples that are heavily contaminated. Okay, so ito po, this picture on the bottom right corner, yan po yung example ng Bacillus anthracis that has been grown on a plant medium. All right, next is on a gelatin stab. So although non-motile po ang Bacillus anthracis, some strains could produce motility patterns on a gelatin stab, or a unique growth pattern on a gelatin stab. So they could produce an inverted pine tree or inverted Christmas tree pattern as shown here in this photo. And then last one, Bacillus anthracis is generally susceptible to penicillin, specifically at 10 units per milliliter. But if we were to grow them in a culture media with only about 0.5 units of penicillin, then we may be able to observe the enlargement of their cells. And this would in turn change their colony morphology a little bit, which would give them a string of pearls appearance, as you can see right here. So mukha daw po silang pearls if they were incubated in a culture media that contains 0.5 penicillin. Okay, next is Bacillus cereus, otherwise known as the fried rice bacillus. So Bacillus cereus is relatively a common cause of food poisoning and opportunistic infections in susceptible hosts. Okay? Side note lang po, yung food poisoning, kahit hindi po kayo immunocompromised, you may still be infected, or you may still be affected by food poisoning caused by Bacillus cereus. So ang food poisoning caused by B. cereus is not exclusively an opportunistic infection. Okay? So food poisoning caused by B. cereus takes two forms. So either diarrheal or emetic sila. So the diarrheal syndrome is usually associated with the ingestion of meat or poultry products, and this is characterized by an incubation period of 8 to 16 hours. Medyo matagal po. Infected individuals have abdominal pain, diarrhea, and vomiting. Fever is uncommon, but sometimes this may also be present. Okay? So ayan, pag diarrheal type po, matagal yung incubation period, and ang primary manifestation niya is diarrhea. The emetic form naman po, it has predominant symptoms of nausea and vomiting 1.5 or 1.5, 1 to 5 hours after ingestion of contaminated food. So compared to diarrheal, mas mabilis yung incubation period ng emetic type. And ang primary symptoms po nito is profuse vomiting. Okay? And so the culture of suspected food items from a food poisoning incident may be done to isolate and quantify Bacillus cereus. So dito po, ang specimen of choice natin, imbis na clinical sample from the patient, ang kukunin natin is food item from the suspected source of food poisoning. So Bacillus cereus can be grown aerobically at 37 degrees Celsius on BAP or SBA. And they would produce beta hemolytic frosted glass colonies as shown in this picture. So ganito na po yung itsura ng frosted glass colonies. And as you can see, kung i-compare natin with Bacillus anthracis, sila po ay beta hemolytic. Ang anthracis, non-hemolytic. Ang cereus, beta hemolytic. Okay. So most cases of food poisoning caused by Bacillus cereus do not require antimicrobial treatment. Kasi po self-limiting naman yung infections that is caused by Bacillus cereus. Usually after 9 hours or approximately 9 hours, ayun po, magsusubside na rin yung clinical manifestations. Ayan, on this slide, special thanks po kay Professor Mary Lirio sa kanya ko po nakuha itong diagram na to. So this is just a simple diagram that could help you identify these aerobic gram-positive bacilli that we've just discussed. So titignan natin kung sila ba ay branching or non-branching, are they spore formers? Tapos ayan po, titignan natin yung mga possible tests na pwede natin gawin to narrow down our choices kung ano nga ba yung mga organisms na pwede nating makita dun sa clinical sample. Okay, so here are the references that were used in the making of this discussion. Thank you so much for listening. Our chat box is now open for questions. So does anybody have any questions?